Okay. Okay, are we ready? Good morning, everybody. You remember last week we talked about very basic principles that control the development of the central nervous system. And today, which is my last talk, my last is as if we had many, my second and last talk, I would like to give you a glimpse on the development of the peripheral nervous system. As I am sure that those who are going to work on the CNS will rarely touch the PNS ever again in your lives. So it's very, very important. These are many, many neurons and many glial cells and many, many functions which are crucial for our body. So at a certain point, there is going to be a certain list that I would like you to remember, but basically the rest is going to be fun, I hope, as we are going to ask about some basic principles. Okay? So let us go back to the issue of neurulation, and now I'm going to complicate it a little bit. So you remember that last week we talked about the ectoderm, the neuroectoderm, and the fact that this neuroepithelium begins folding upon itself until it closes into a close structure. And this is, I believe, the picture that I showed you last week. So how the neural folds are approaching each other in the midline, and at the end of the process, they are going to fuse with each other and create a closed neural tube, whereas the ectoderm that is going to become the epidermis, okay, is going to regenerate dorsal to the entire system, okay, or above, if you wish, a more intuitive word, the entire system. So this tube, as we already know, is going to develop into the entire spinal cord and the brain, but, and now come the news, it is also going to give rise to a population of transient progenitors, which are termed neural crest cells. And these neural crest cells have a transient lifespan. They do many things about which we are going to talk, and then they will generate most of the peripheral nervous system and many other derivatives. So this is going to be the issue today. So if now we make a section, namely, again, we have the baguette, and we slice the baguette, and what you see here is a slice, okay? We see the sliced stuff, what we call a cross-section through the tube. And what you can see is the neural tube, the notochord, a structure which will become important in a few minutes, the mesoderm, okay, that we talked about also, and this is the ectoderm, which is going to be the epidermis. I am very repetitive, not because I don't remember what I said a minute ago, but because I believe repetition is important for you just to integrate things, okay? So bear with me on that. If you don't like it, just tell me, and I can run a little bit faster. So why do I show you this? Because at this particular time point, the neuroepithelium, histologically speaking, is a pseudostratified neuroepithelium. We already mentioned this term, okay? In which the entire extent of the epithelium is composed of one cell, which cycles, and its nucleus is at different apicobasal positions based on the cell cycle time in which they are located. But look at this area. This is where the neural crests develop. And what you see here is an in situ hybridization, a reaction RNA-RNA, with a transcription factor, never mind the name, I'm not interested to complicate you with names, but this is a very important molecule that is already expressed in the what we call pre-migratory domain of the crest, namely in the dorsal, this is ventral, in the dorsal neural tube. So this means that although at the cellular level the entire neural tube is homogeneous, it is only apparently homogeneous because molecularly speaking, we can already identify domains of heterogeneity, of molecular heterogeneity. And what does this mean? Not differentiation. Nothing is differentiated at this point. Something a little bit more subtle. Right. So there is already a kind of specification, a kind of differential identity to those cells that are located here when compared to those cells that are located at more ventral positions. And in fact, it is known that the specification to become a neural crest cell occurs even before the closure of the neural tube. And there are people who claim that it already occurs at gastrulation, a stage that we are not discussing in this talk. So what characterizes the neural crest? The neural crest is characterized, and this is that was the crest at the spinal cord level, and this is the crest at the mesencephalic level, at the midbrain. So you see the neural tube, and here you see that the population of crest cells, instead of remaining in the CNS, is going to leave. So the first change, the first characteristic of neural crest cells is that they are born within the epithelium, but in order to perform their task in life, they have to leave the neuroepithelium. And they do so by a process which is called epithelial to mesenchymal transition, okay, that we already saw and that I'm going to show you an animation for that. So at the mesencephalic level, the crest cells live in a very special manner. They don't undergo a full EMT, a full epithelial to mesenchymal transition. They first leave, and only then, before they disperse throughout the embryo, then they undergo full mesenchymalization. So we are talking about a population which is born as an epithelium, undergoes EMT, and later on, and I'm not going to show you that, migrate as single cells, as chain of cells, throughout many, many stereotypic pathways in the embryo, through the mesoderm, between blood vessels, underneath the epidermis. And then they reach their final target sites where they stop migrating, begin organizing, for example, into ganglia, and then they finally differentiate. So we are talking about a system which is one of the most studied systems as models of cellular migrations and movements within the embryo. Now, since they are not the first cells to be created in the embryo, the mesoderm is there, the basic network of blood vessels is already there, a lot of extracellular matrix in between the tissues, crest cells take advantage of those pathways to migrate in stereotypic manners. We have characterized, like 20 years ago, most of the migratory pathways of neural crest cells at the level of the trunk. Others have done so at the level of the head, and these pathways differ at different levels of the axis. Okay, in the tail it's different, in the trunk it's different, at vagal levels, the neck, and of course at cephalic levels. So let me show you now how it looks in the trunk. You can see the process of EMT or delamination. This is the tube, and look at these two pearls here, like two tears that we can see. These are crest cells that have left the neural tube. This is an isolated preparation, and you can see them before they are beginning to disperse. Let me show you now a movie of one of my students who, what she did is she electroporated with GFP encoding DNA the neural tube. Then she took the neural tube out on a dish, and what you see here in this part, okay, are cells that have been electroporated with the GFP, are still epithelial, okay, and now they will begin emigrating, and this is what they do. Okay, so these are live cells, true cells of avian embryos. And look at the conformation of these cells. They are mesenchymal, they are stellate. You know, they have like philopodia and pseudopodia and all kinds of podia that enable them to sense, if it is a dish, the substrate, which is fibronectin, and then they can advance, okay, by putting up a process, retracting the rear end, and then crawling on the substrate as mesenchymal cells. And this is what they basically do in the embryo as well, but in a 3D context, or 4D context, I should say, because obviously it is time dependent. Ken? Okay, in the dish, what causes them to move is the movement from high density to lower density. Basically, they sit on fibronectin, as I said. Now, crest cells express fibronectin receptors as low affinity receptors, which enable them to contact the ligand, respond to the ligand by changing the properties of the actin cytoskeleton, and since the receptors are of low affinity, they can move, they don't stick to them. Okay, so this is how they do it in the dish. Now, in the embryo, can you imagine how they move in the embryo? So this is the process of EMT, okay? This is the epithelial cell, and this is how they mesenchymalise. We spent 10 years on this project in trying to understand how EMT takes place, and now we have a molecular network that I'm not going to tell you about, we know is responsible for the onset of emigration of neural crest cells. So how, how do you imagine these cells migrate as they migrate in vivo? Okay. That's right. Here there is no chemical gradient. There is a substrate. Okay, so they cannot move from here to another place. They have to move to a free, empty, cell-free space. But in the embryo, some of the guidance cues are basically chemoattractants or chemorepellents.
It doesn't seem like they're doing like random movements that would cause them to spread.
No, that's right, because there is contact inhibition of locomotion due to cell contact. Because here they migrate as single cells, but if you will see it again, you will see that they create transient contacts with each other. These contacts are mediated by cadherins, which cause what we called CIL, contact inhibition of locomotion, and this puts them along a certain pathway. So if it was one single cell, then its movement would have been random. But since they are in cohorts of cells, the movement is not random. And this is what happens in vivo as well. So they migrate along gradients of positive and negative signals, and they also interact with each other, showing in vivo also contact inhibition of locomotion, and this brings them through different pathways. And there are obviously certain limitations due to the properties of the different pathways. For example, cells migrate. I can show it to you here. Certain cells will disperse underneath the ectoderm. So this is not empty. This is full of mesoderm, okay? This is histology, so you don't see it. But this is mesoderm, which at this point cannot be penetrated by crest cells. This is ectoderm, which cannot be penetrated by crest cells because there is a basement membrane underneath. So the cells will disperse here because they don't have any other choice. And there is a lot of proteoglycans, which are hydrating molecules that will enable the cells to move through this space. And this type of constraints generated by the adjacent tissues will somehow dominate or control the pathway through which the cells are going to move. And these pathways are dynamic. They change throughout development and throughout migration. So each one of the things. EMT is a project on its own. Migration is many, many projects because it changes throughout the axis and so on and so forth.
Do you consider what you showed in the dish type of migration?
Of course. Yeah.
I mean, how do you define migration?
Migration, the movement of cells from the source towards a certain target. In vivo, in the dish, there is no target, so you define it as the onset and progression of movement.
And do you have such mechanisms that they move because of, something like concentration gradients and stuff?
You can do it in vitro too. I'm not a fan of in vitro, don't... okay. Of course. Of course, you see in vivo so many mechanisms. In different areas you have different mechanisms. Right. You see everything. You see cell contact, you see limitations by adjacent structures, you see repellent mechanisms, chemoattraction, permissive mechanisms, and just movement of groups of cells. Everything exists in different areas of the body. Additional questions? Okay.
Is EMT just a migration process?
No, EMT is the generation of a cell that is able to migrate. Is the transition, the conversion of a cell which is epithelial and and static into a cell that is able to move. Then the next stage in the process is the beginning of migration. So you can absolutely separate emigration, delamination, okay, with migration proper. Migration takes sometimes a few days. Emigration is a shorter process. Again, depends on the species, on the area, and so on. Okay, is this difference clear? So, as I said, because of these 2 hours, we cannot cover all the topics that characterize the neural crest, which is a very, very hot topic in developmental biology and neurobiology. And now the famous table. So cells have migrated. For certain reasons, they stop migrating. We are not going to enter into this topic either. And then they begin differentiating. They begin coalescing into ganglia, mainly, but not only, that will form the peripheral nervous system. And then I'll tell you why I'm saying that is most, but not all, the peripheral nervous system. Remind me if I forget. Okay, so let's go through this table. It's important. Boring, but important. I'll try to make it less boring for you. So, peripheral nervous system is formed of sensory ganglia. Right? What is a sensory ganglion? What do you know about sensory ganglia? The biologists among you. Okay, and what are the modalities that sensory ganglia mediate? What do we sense through the sensory ganglia? Mama? Can you be more specific? What else? Proprioception. Have you heard about this? I mean, the sense of my being in space, okay? So touch, pressure, pain, temperature. Okay, all those are modalities which are transmitted through the neurons of the sensory ganglia. And this is why a characteristic sensory neuron is bipolar. We'll have one neurite going to the periphery and the other one to the spinal cord, of course, or to the brain, right? Because it has to transmit information from the periphery. A first transduction takes place in the sensory neuron within the peripheral ganglion, and then it goes towards the dorsal portion of your spinal cord, mesencephalon, diencephalon, or whatever. Okay, so these are sensory ganglia, and sensory ganglia can be classified in 2 main categories. Sensory ganglia of the trunk, which are called spinal ganglia, or in other words, dorsal root ganglia. Have you heard this term? DRG are all the ganglia located on both sides of your spinal cord. And then you have what we call cranial sensory ganglia. These are a bunch of sensory ganglia located in your brain, outside the brain itself, but attached to it. Okay, while outside, because it's PNS. While outside, because the cells underwent EMT, right? So we are outside the CNS, but with connections to the CNS. And examples are the trigeminal nerve, or the trigeminal ganglion, of course, the facial root ganglion, the glossopharyngeal, the vagal, and so on and so forth. The jugular, we have many. Okay? So why did I tell you that not all the peripheral nervous system comes from the crest? Because of the cranial sensory ganglia. In the trunk, the dorsal root ganglia contain neurons of different types, which transmit these different sensations. They are transmitted by different categories of neurons. I'm not going to enter into it. Okay, but the neurons, as well as the glia or satellite cells of the ganglia, all derive from the neural crest. However, in cranial sensory ganglia, there is a mixed population of cells, and I'm going to draw this for you. I should have erased this. So what happens in the brain is the following. In the brain, This would be the brain, okay? And the ectoderm. You have two populations of cells. You have the neural crest cells, which live, but you have another population, which is called placodal cells. These placodes, like those which will generate the ears and your eyes, are thickenings of ectoderm. It is like the ectoderm would create an ectopic little mini brain here. These are called ectodermal placodes. And these placodes will generate cells by delamination as well, by EMT. So cells from the placodes will join cells from the neural crest, and all of them will give rise, for example, to the trigeminal ganglion. I'm giving you an example. So in the trigeminal ganglion, you have large neurons, small neurons, and glia. Now the large neurons come from the placode, and the small neurons and glia come from the neural crest. So this is what we call a mixed type of ganglion, and this is why I told you that the crest forms most of the peripheral nervous system, but not all of it, because the placodes in the brain, and only in the brain, have an essential contribution to the formation of neurons. Let me give you a little bit more of a hint. These large neurons in the trigeminal ganglion are going to be proprioceptive neurons, responsive to neurotrophin 3, will express the neurotrophin receptor tract C. And just if I'm hitting anybody here, if you don't know, forget about it. Whereas, for example, the small neurons are going to be pain-transmitting neurons, nociceptors, and they come from the crest. And evolutionarily speaking, this is older than the pain-transmitting neurons. This has been shown in evolutionary studies, as if the pain sensation would be newer in evolution than proprioception or touch or pressure or stuff like that. Okay. What?
Where is from which part of
The placode?
Yeah.
From the ectoderm. This is why we call them ectodermal placodes. Okay, so you take about, the ectoderm has thickened to form the entire tube in the midline. It thickens also on both sides, only in those parts of the brain. So, this was the reason, and most of the ganglia, glossopharyngeal, etc., are formed in such a way. In most of them, all the neurons are placodal and all the glia is crest. In the trigeminal, it is mixed. So it's a nice example of a mixed type of ganglion. Okay, so sensory ganglia. Next, satellite cells. Satellite cells is what we call glia. Satellite cells are the glia of the ganglia. And they all come, in all types of ganglia, they come from the crest. Then we have something extremely interesting, which is the autonomic nervous system. What is the autonomic nervous system? Read and tell me. Explain me a little bit more. What do we know about the autonomic nervous system? מה שאתם יודעים, whatever you know, will be helpful to set the scene, to let me elaborate a bit more. Okay. Anything else? Correct. Anything else?
That's voluntary. Freedom is voluntary.
Is it sensory or motor? No, it's motor. It's a motor system, right? Okay, it activates. Okay. So the autonomic nervous system can be divided, as it is written here and it was said, into sympathetic ganglia and plexuses, parasympathetic ganglia and plexuses, and something which is of absolute interest, the entire innervation of your gut. From your mouth till your anus, everything comes from the crest. All the plexuses, all the neurons, all the glia, everything comes from the crest. And the number of neurons and glia and the variety of neurons is similar to the spinal cord. Okay, many people study this. It's less regarded. Many things have been discovered during the past 10 years or so about the enteric nervous system. So, the sympathetic ganglia and plexuses, obviously, okay, innervate muscle, innervate sweat glands, innervate your heart, innervate your gut. So, the enteric nervous system sits within your gut, and the sympathetic and parasympathetic systems control your gut, control the motility of the muscles of your gut. Okay, whereas the sympathetic system increases peristalsis, the parasympathetic system will make it a little bit relaxed. And the same is for the heart and for sweat glands. If you perspirate less or more, if you run to the bathroom before you have an exam, all this is sympathetic and parasympathetic systems. And now I'm going to address the Schwann cells of the peripheral nerves, okay? Namely, all the cells which are myelinated in the periphery, which leave the spinal cord, for example, as motoneurons, or the neurites of sensory neurons, or of autonomic neurons, all of them, or some of them, the pain ones will not have it, will have myelin. Right? You know what myelin is. So the cells responsible for producing the myelin in the periphery are called Schwann cells, and they all come from the crest. So this is an additional derivative of the crest. In the CNS, which cells myelinate? Oligos. So they are different, distinct in origin, etc. Now I'm coming to the fight and flight stuff. It appears that many other derivatives are generated by the crest. I'm not going to talk about those ones. I'm going to talk about chromaffin cells. These chromaffin cells are located within the adrenal medulla. The adrenal glands are above your kidneys, bilaterally located. And basically, they are composed of a cortex, which is mesodermal, and a medulla, a liba, an inner part, which is composed of chromaffin cells that come from the crest. Now, these cells are believed to be, and we demonstrated that a few years ago, to belong to the same lineage as sympathetic neurons. Namely, when they leave the neural tube, a single cell is able still to generate a chromaffin cell and a sympathetic neuron. But then their pathways diverge, separate. Some of them will colonize the ganglia, sympathetic ganglia. Others will run up to the cortex and generate the medulla. And then they will become hormone-secreting cells instead of neurons. And these hormones, which are corticosteroids, will mediate the fight and flight response. Namely, I see a fearful stimulus and I fly away. I have this very primitive and absolutely necessary type of behavior, okay, which is mediated hormonally by chromaffin cells, which are crest derivatives. So you can see already that there is a high variability of cells that are generated by the crest, and this is not all. Now I'm going to talk very briefly about two different types of cells, which nobody understands up to now why they are generated by the crest, because the crest is the neural crest. So it appears that the entire pigment of your body, all the navy we have in the skin, if you are black, if you are, I don't know, from another race, the beautiful colors of the birds and of fish, all of it is neural crest. So now when you wake up and you look through your window, you see a beautiful bird, you will see neural crest. And this will be true. You'll see a few pictures later on. So the crest will generate the entire pigmentation of the body except for the retinal pigment epithelium. Okay? You know that in the retina there is a layer which covers the retina, the neural retina, which is called RPE, retinal pigment epithelium, and this comes from the diencephalon. This comes from neural tissue of CNS origin, not crest. And this is a very important evolutionary thing. Again, I won't have much time to talk about it. It is believed that pigment cells and Schwann cells have also a common origin in the pre-migratory crest for some reason. And there are many, many diseases. For example, schwannomas, cancers of Schwann cell, or tumors of Schwann cells, not necessarily malignant, in which you can see pigmentation. They are problems of depigmentation in people with schwannomas, and so on and so forth. So there is a linkage between these two sublineages of the neural crest. And last but not least, I'm going to speak about this a little bit. Forget about the detailed names. But what I would like to claim is that the cephalic crest, and only the crest at the level of the head, will generate, in addition to neural structures, also what we call mesectoderm, which is a very weird word. Why? Because it has a mixture of mesoderm and ectoderm. Why is it so? Because look at this. We are talking here about bones, about dermis, and about some facial muscles. So all this in the embryo is normally generated by mesoderm, by another germ layer, not by ectoderm. Ectoderm, we have been raised saying that ectoderm generates your skin and your nervous system. So how come that the crest, an ectodermal derivative, will generate bones? And I'll tell you now the theory. Of course, it's hypothetical, but let me show you which bones. So this is a chick embryo, chick, not an embryo already, and what you see in red, of course, this is the beak, okay, maxilla and mandibula. This is the calvaria. This is the eye, and these are the orbit, okay, all these bones around the eye. So basically it would be this area here, okay, up to what we call the basis sphenoid, in this area there, okay, and all this is neural crest. And only these blue bones will come from the mesoderm of the head. So it appears that the crest contributes to bones, and it appears that in human, this would be the equivalent. So if you see a poor human being with its brain, what you see is maxilla and mandibula, and again, the entire covering of your brain, except for the basi-occiput and another bone here, which comes from the cephalic area. So it appears that the crest, only in the head, will generate mesodermal derivatives, such as bones. And it also generates dermis, etc. And now the theory. How does this take place? So there is a beautiful paper, very old paper, which is a hypothetical paper by Gans and Northcutt, which claims that the neural crest has basically, or is basically, an invention of vertebrates, which moved from an aquatic environment towards a terrestrial environment and had to develop predating capabilities. These predating capabilities transformed the head into something which needs, okay, to predate. You need to have a very strong mouth, a maxilla and a mandibula, able to predate another animal. And this has, in fact, evolutionarily evolved, okay, with the transition into a terrestrial type of life. Now, it appears, and now I have to do a little drawing. It appears that your notochord, you have the neural tube, and then you have the brain, okay? And we know that the notochord is localized underneath the entire neural tube, but it finishes at a certain point here, around the diencephalon, mesencephalon, diencephalon border. No more notochord. What is the meaning of no more notochord, says this hypothesis? You remember that I told you last week that the notochord secretes sonic hedgehog. And it secretes sonic hedgehog in a manner that sonic reaches the neural tube to create a gradient that generates motoneurons, interneurons, and so on and so forth. But this very same sonic is also able to radiate towards the mesoderm. And in the mesoderm, it will create your vertebra and also your muscles in a gradient. But it will be basically the factor that is important for specification of ventral somatic cells into vertebra. Namely, you will become a bone thanks to an initial input by sonic hedgehog. Now, if, this is what the theory says, if I have then sonic all along the axis, this means that all the bones in my trunk will be of mesodermal origin. But if this stops here, and there is no more notochord, no, in the area of the brain, something else has to take over the function of the creation of bones. Why should we have bones in the head? Because during the process of the transition of vertebrates to terrestrial environments, we had also a very important process that is called cephalization. The size of the brain tissue, has increased enormously. And so, obviously, all the senses that have become centralized to the brain, eyes, ears, olfaction, touch we have everywhere. But look at this. Evolutionarily, all kinds of worms, they have their sensations everywhere throughout the body. But we are centralized animals. So there is a centralization of the senses. There is a very, very rapid development of the brain, which is not optimized, by the way, in terms of gene expression and so on. Okay? So this brain needed a kind of cover to protect it. And this cover has to be osseous and cartilaginous, of course. And in the absence of sonic, which would induce the cephalic mesoderm to produce this coverage, the neural crest took over. This is the hypothesis. I'm not going to tell you the molecular underpinning of this hypothesis. Okay, took over to create this osseous environment that protects the brain. So for more reading, you are all sent to this extraordinary paper. Right, 1 second. So, transition between protochordates and vertebrates, acquisition of skeletal tissues, development of a head and a brain, a very, very, very important process of cephalization. Okay, now we have a brain. Lower animals do not have a real brain with sense organs and jaws as organs of predation. So the crest undertook this function of creating the jaws and protecting the brain. Right, Ken? Questions? I'm not sure I can answer all of them, but questions.
Fish do have skull, right?
Fish are vertebrates.
But you said it's when we let the things
Right, but fish are cartilaginous. Most of the, you have bony fishes, etc., but most of the covering of the brain is cartilaginous. Is not osseous. Okay, but of course you have, yeah, but you obviously have in fish. By the way, it hasn't been that determined whether the osseous or cartilaginous covering of the brain is crest derived or cephalic mesoderm derived. But they certainly don't have jaws the way terrestrial animals have. Okay, you have. I think that the ancient fishes are cartilaginous fishes, and the more modern ones are bony fishes. But bony in terms of their skeleton in general. yeah, yeah, no doubt about it, but this is a chain in evolution. You don't have a transition which is sharp. You have a very smooth transition, okay? But the real transition, okay, takes place there. But we are talking about vertebrates, and fish are still vertebrates. But the formation of the jaw as an organ of predation is a terrestrial innovation, more than in fish. More than in fish. You have, of course, predating fish. I'm not saying that, I mean, this paper is not saying that this happens, in a sharp way. This is an evolutionary process. So it has to be present in certain maritime or aquatic animals as well.
And the protocol doesn't go all the way
No,
along
no, no, no. And in fish, this is a very interesting thing. In fish, the form in which the neural tube is created is very different from the way in which it is created in our body. So many things are different. I mean, this is a theory, right? Very, very difficult to prove, but it makes sense because it is an association. I mean, okay, you have crest in fish too. No doubt about it. But for example, as I told you, I think last time, a dorsal root ganglion in the zebrafish has 3 cells, 3 neurons, and a couple of glial cells. A sympathetic ganglion may have, 5 neurons. That's all. So everything is predetermined there. It's very different. It's a kind of transition type of animal between invertebrates and vertebrates in terms of crest. Okay? It's very, very complicated. Additional questions? Where are we? Okay, let's take a break maybe now, and then I would like to move to a little bit another topic. Okay? זה בא מהגיל, המפורסמים האלה, אוקיי? חלק מהגיל אנדוקרינים, אבל לא את כולם. למה?
זה נורמה רגילה. אתה יכול לבנות נורמה דואלית שכל נורמה. הנורמה הדואלית, הנורמה הדואלית, הנורמה הדואלית.
זה מקיים את מה אתה חושב מה שקוראים לו דואל.
בדיוק. זה מקיים את מה שאני מטפל עם דואליות. זה כזה, יש כל מיני נורמות דואליות מוכרות. זה כזה, הנורמה הדואלית של L2 זה עצמה, הנורמה הדואלית של L1 זה כסף של L1 זה L2. זה יותר כאלו. זה יותר גדול ממה שאנחנו מגדירים בתוך רגיל, אבל זאת ההגדרה של נורמה דואלית, בהינתן איזושהי נורמה, אבל נורמה דואלית פשוט מקסימום
המקסו על Y.
המקסימום על המקום, כאילו, אני רוצה שזה יהיה
האופרטור מקס מתקדם על Y, כי X הוא...
X זה לא פעם, אתה בוחר Y ככה ש...
Maximize Y such that Y קטן.
אני לא יודע הגדרה של נורמה, ואז כאילו, שאני אוכיח את הדבר הזה, זה איזושהי קצת, מה אתה מנסה בעצם לעשות? מה אם הוא חזק משני האגפים? כאילו...
כולם מתים לעוף הביתה אחרי שבוע שלם של בחינות, עובדים ככה על
תנסה לגרום לדבר הזה להיראות בגדול כמו שורש XPA מינוס 1. כאילו זה נגלה הערך המקסימלי פה הוא בעצם אני מנסה ש-Y
איזה קורס?
הוא אמר לי לי כבר
כן, נכון.
פסיכיאטריה.
זהו, אבל כאילו את מצפה
ש-Y יהיה זה מעניין. איך זה מעניין? לחלק ולהכפיל בוקטורים זה
לא עובד.
אצלנו זה עכשיו קורס ממש טוב.
כן?
כן.
אני יודעת שתקראו אותו.
מה? לא. את הסיבה שלו. רציתי לעשות, ולכן אני חושבת.
למה היית בזה? בזה נגולה? אתם הייתם פה?
כן. יכולנו לדבר בר אילן.
כן. תקראו אותו, הוא היה לו כל מיני סיפורים.
מה? של המקודן?
אמרו שהיה לו רומן תלמיד.
הנורמה של תלמיד, הנורמה A במינוס 1.
זה לא יותר גרוע מסיים תלמידה.
לא. חד משמעית לא. זה גרוע נקודה.
כאילו יש לך איזה זה ככה פה, כן.
אפילו בשנה האחרונה שאתה חיידה.
לא, לא תלמיד נקסט. זה היה מישהו הוא היה בתל אביב. זה מעצבן. כאילו אנשים שנה מעליהם כבר, כאילו, בסצנות, היו באים ואומרים, וואו, זה כל מדהים, הוא כל זה, הוא פה ושם.
מה הוא עושה עכשיו?
התחלנו מכזה, הוא הוכיח שבעיה הייתה איזושהי בעיה הייתה אם אפשר לפתור את הבעיה הזאת בפחות מ-n בריבוע אז פעילות הבעיה תראה את זה כזה כאילו משהו יותר חזק מכאילו
אבל אז אני עוברת ממטרון, נגיד, אתמול נסעתי
אבל זה קרוב סמטר טיים
ועברתי במסדרון
זה היה אחר
ואנשים עושים כאילו עמוק בפנים בעבודה בשמונה בערב.
אתה יודע מה מוזר? שכאילו יש פה איזשהו...
אני אתמול קניתי נעלי בית בצורה
אולי אפשר
יש פה איזה עבודה עם ה...
ואני כאילו ישבתי
כאילו עם המין הנורמה הקנונית באיזשהו מובן. אם רק קרובה...
וואו.
כן, נכון. זה כאילו... אז הקנוניות הזאת, כאילו... אם אתה משנה את הבסיסים איכשהו, כל הדברים האלה אמורים לנבוא. כאילו, אני חוזר למה שהייתי אוהב להגיד. אבל זה לא חושב, לא שתיים. יש איזה אקטיב, העלותות של האקטיב. ואז המקבילה הפנימית, לא, אבל כל משבר פנימית, אתה יכול להגיד כאילו אוקיי, אבל איך אני עושה? אני לא יודע לא יודע. זה ה... לא יודע. רציתי לשאול כמה... בתור התרבות שלי משפט.
תמיד תעמדות לחלופות. פרקטי, שם פרקטי. כן.
אני בדרך כלל שאני טיפה סדרה,
קורה אם, אוקיי, פה פתחת
כל פרק זה 40
את זה, מה קורה אם אתה מתחיל מהצד השני? זה לא מלא.
זה לא מלא, אבל זה כזה...
אז כאילו, זה לא גם בטוח לאן יש לך להגיע חוץ בלי זמן ולכל את
גם
הלאה.
האח שלי רואה את זה, והוא מסיים את הספר בשנתיים כנראה זה ממש ספציפית. יש שירים טובים, תראי את זה רק בשביל המוזיקה, יש שם, לא יודעים, מצלמת, תראי את זה רק בשביל איזה מה, תראי את זה רק
כאילו פה יש לך את זה כאן, אבל אין כאילו גם אם עושים פעם להגדיר, אבל לא היו אבל זה מתקפל מגזים, זה לא מתקפל כמו נורמה.
וואי, אני כל כך את משרד הרשם. תזכרו
Okay, can we continue? Are you spiritually ready? So what I would like to do in the 45 minutes or so that we still have is to show you some embryology, classical embryology that paved the way to the knowledge we have, and most specifically to the table that I gave you of derivatives. And second, to deal with you about the very, very basic issue in developmental biology in general and neurobiology in particular of how or what is the state of commitment of a cell, naive, specified at a certain point in development, and how do we approach the study of this very big question. Okay? So, in the 70s, there was a woman, she's still among us, called Nicole Le Douarin. I was a postdoc in her lab, and she made a seminal in Paris, which was fun, and she made a seminal observation. She looked at nuclei. Let me begin by another slide. She looked at nuclei in two species of birds. She looked at how do nuclei look in chick embryos and how nuclei look in quail embryos, slavim, for those who, okay? So she says in the quail, the nucleus is organized, or the DNA is extremely compact and is organized in chromocenters within the nucleus. Whereas in the chick, it is dispersed. So using this technique in the 70s, now we have antibodies that recognize quail. You will see it in a second. So now life is easier. But then she says, if I have two species of birds that are different in terms of their appearance, cellularly speaking, then I could combine or graft the neural tube from a quail into a chick embryo instead of the tube of the chick embryo. And when the crest cells leave the neural primordium, I can mark them and I can follow them at each and every time. Because cells having the quail marker will be distinct and distinguishable from those of the host. And why did she have to think about it at all? Because when crest cells leave the neural primordium and they migrate through the different tissues, they are not different from the other tissues. We cannot recognize them. At that time, when this discovery, this table was made, we didn't have any idea of molecular markers of crest cells, not of SLAG and of FOX and of SOX and of the many receptors and things that are trivial for us today. So we had to find, she had to find a way to follow the cells in a manner that does not alter their normal development, okay, and that will enable us to follow them from their exit from the neural tube until they form the ganglia, and even more, until they hatch. Why not? And this is what she did. So what she did, this will be enough. What she did is she took a chick host. This is a 2-day-old embryo. Okay, you see the head, the encephalon with the eyes, mesencephalon, rhombencephalon, the somites, the spinal cord. Okay, these are the omphalomesenteric veins. What else? This is the heart. So, and this is the ear, the otic vesicle. So what she did is she removed the spinal cord or the neural tube at this stage from the chick host. So she left a hole. You see it here? Nothing. This is the notochord. And in the place of the space left after the removal of the chick neural tube, she placed a tube which comes from a quail donor. Now, the tube remains in place. But the only cells which leave the tube to migrate through the neighboring structures are the crest cells. So she is able to follow the crest cells during migration and during the formation of all the organs. This is the way in which the table that I spent so much time last hour was created, via chick, quail, chimeras. So she was able, with additional markers, of course, to determine which neurons, which glial cells, in which places, etc., are coming from the crash. She did the entire mapping. You'll see it in a second, what this chimera will become. So when doing this, she did it with fragments of tubes, and she did it in the tail, in the trunk, a little bit here, 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 up to the head. So this was a mega project in her lab, which lasted for many years, because she basically mapped the fate of every area of the axis. And this is not trivial. I learned to do it. It's beautiful to do microsurgery within the egg, because you open an egg after you incubate it for 2 days, and then you put a little bit of pelican ink underneath the blastoderm, because the embryo is completely transparent at this stage. So in order to see what you do, you put a little bit of Indian ink, which is a suspension, a black suspension, and then you begin removing. Whoever wants, I can show you a movie of how we do it. And then you remove the membranes, and you're facing the embryo with the ectoderm, and with microscalpels, which have a micron sharp tip. You can perform microsurgically this procedure. No enzymes, no nothing. And you can do it. To remove from the host, from the donor, you need enzymes, but not to place it. And this is how you can do it. And this is basically an example. For example, here, now that we have already an antibody, okay, instead of this is called the Felgen technique. It's an histological type of technique. Now we have QCPN, quail, whatever, and here you can see. What is this? What did we transplant here? Obviously not the neural tube. What did we transplant here? Huh? A somite. You remember the somites? These cubes on both sides of the tube? So this we did for another project. We transplanted a quail somite. Instead, this is the chick one, and this in the other side is the quail. So it's beautiful. It gets integrated. It develops completely normally.
How can you, it's very difficult question, but how can you make sure that you only take like a single type of cell existing in that area, and you didn't take a little bit of mesoderm?
This is, first of all, because you see it in your result. Very obvious. And second of all, because the neural tube is separated from the mesoderm by a basement membrane. And it's very, very easy to remove it properly. But people who are not experienced will take mesoderm along with it as well.
How can you control the size that's fully on one side and on the other?
Because this is what you do. You remove on one side and you put in one side. You have complete control of what you do. It's very easy. Easy to say, more difficult to do, but you can do it. It is doable. Many laboratories still do this. Now we combine it with a lot of molecular biology. And this is what comes out from this type of grafting. So you can see that these embryos are able to hatch, and they are normal up to 10 days of development, because only at 10 days the immune system is developing. And then they begin reacting against the graft and creating a kind of neurological disease called, it's like a myoencephalitis, okay? The T cells and B cells begin attacking the graft, and these animals will eventually die. But it is also possible to prevent this immune disease by co-grafting a piece of thymus the same day in which you do your neural tube graft. And this is something I know one person in the whole world who was able to do it. Okay, and she's already retired. So it is possible to prevent the immune rejection and to get these animals to be adults. Okay? If you do a co-transplantation of a piece of thymus, which mediates cellular immunity. No, you do this 2 days later. It's absolutely delicate, because there is a lot of blood already, and the embryo can die because of hemorrhage. I'm telling you, this double transplantation, I know one person who did it and not more. But this was to make a principle, okay, not important for us. So what you see here is the white chicken. This is the Japanese quail. Okay, it's quite an aggressive animal, I must say. When we were in France, we had a house. And this was a house which was like 150 years old. And they have what is called in French un poulailler. So they had cages with chicks and all that stuff. I mean, only the cages that when we rented the house. So at one particular time, I decided that my children had to see some birds in the garden. So I brought eggs and we put them on the chauffage central. We put them on the on the, you know, like the oven, like the no, what? No, on the radiators which heat the house, and they hatched. Okay, and then we had chick and quail eggs, and we had a lot of quails in the garden. I put them in cages after 1 week, and it was then that we could define which was a male and which was a female, and the female killed the males. I mean, that was horrendous. And the males ended up with a nuchal hole, all of them the same way. I don't know how, I don't know why, but since then I don't like them. Also, we continue working with them every single day. We get per week like 600 eggs of this kind. We have a person who grows them for us exclusively, and we pay a fortune for them. So these are the Japanese quails. These are the chicken, and as you can see here, these are 3 chimeras. So how do I know they are chimeras? The pigment, because I told you that the crest generates pigment, and you can see here, okay, that this is a chick, but the trunk was replaced with a neural tube from a quail. So the crest resulting from the migration gave rise to pigment, and we know these are chimeras. Okay. They continue. You get enormous tarnegolot, enormous chickens, normal chickens. Yeah. Okay, so my point was not to show you pretty pictures. My point was to show you what was basically the result. And the result, as I told you, was this table that I showed you before, showing you the technique. And I am a little bit surprised that nobody asked me, how did we get to this result? How do we know that? But in fact, what she found, she and others, found that eventually mesectoderm, what I told you, was confined only to the head, that sensory ganglia were produced in the head and in different areas along the axis. Okay, it appears basically that there are almost no sensory ganglia in the tail, by the way. The last area of the tube does not generate sensory ganglia. It generates glia, it generates melanocytes, almost no sensory ganglia. And sensory ganglia begin to be formed, the final ones, only from the 5th or 6th vertebra. Why? Because they are generated all along the neck. However, the somites in the neck will dissociate and disappear. And so will the ganglia. These are special ganglia called Frorieps ganglia, which are created normally and then they die. Very interesting evolutionary thing. Parasympathetic ganglia are located either in the tail or in your head, as you all know. Sympathetic ganglia are located along the trunk, not in the head. And enteric ganglia are very interesting. Enteric ganglia, those that innervate your gut. Enteric ganglia, it appears, let me move this. It's standing here for a week already. So enteric ganglia, this is very interesting. Look at the gut. This is the gut. Esophagus, stomach, duodenum, intestine, which is not curved yet, okay? This is the cecum, something which is the area of our appendix, and rectum and anus, let's say. Okay, so the gut appears as a tube, pretty much like our neural tube. Initially, it's a tube before it begins to be convoluted and complicated. Most of the most of our organs are tubes. The heart is a tube. Okay, it's very interesting. But this is a course. Come to the medical school to listen to the embryology course for medical students. So this is the gut, and the innervation of the gut comes from two areas only. One would imagine that the entire length of the tube will give rise from here, cells that will go to the gut. But this is not the case. It appears that there is a contribution of the vagal area, which is the neck crest, and from the sacral area, which is the crest that comes from the tail. And the vagal crest will migrate through the embryo dorsoventrally and colonize the entire length of the gut, from mouth to anus. And the sacral crest, however, will colonize only from the umbilicus, namely from this area here, up to the sacral area of the gut. So this area here will have a double contribution of cells coming from your neck and from your tail, which is interesting. Nobody knows why this is the case, but we know of many diseases, and in particular one disease which is typical of Ashkenazi Jews. Also I know a few Arab families, which is called aneural megacolon, or Hirschsprung's disease. I also give a mini course on that, in which crest cells come out. However, they migrate the length of the gut, and they stop here. They don't migrate more than that. So this area remains aneural. So why doesn't the sacral crest generate most of the neurons there? Because it appeared that the sacral crest generates only a minority of neurons. Most of the contribution of the sacral crest is glia. It has been demonstrated that they give rise to neurons, but very, very few when compared to the millions of neurons that are generated by the crest coming from the neck area. And in these children, they are born without crest that innervates their lower intestine. Sometimes it's transverse and descending colon, sometimes it's only descending colon, sometimes it is only the rectum. So it depends on length, and therefore it depends, or it will change the severity of these cases. It's 1 in 5,000 or something, quite high, the incidence. And these children will be born with a balloon of an intestine. So if the intestine has to undergo peristalsis and has to be able to obviously contract the muscles to move feces, the intestines of these children will not be able to do that. And therefore they balloon. So they have the normal gut and then a balloon. So what they do with this, many children have very bad prognosis. In other children, what they do is they cut the damaged part of the intestine, and if it is not too long, they will fuse the two distal and proximal parts, or in other cases, less fortunate, they will add a sakit, they will add a bag, an external bag, into which these children eventually will defecate. Okay, so these are all problems of the neural crest, problems of innervation of the system. So this is what we call a fate map of the neural crest, namely what kind of derivatives does the crest generate at each level of the axis, and they are various and different. So this leads me to the last point I would like to discuss, which is relatively long, which is the lineage analysis of the crest. So if we see this structure, and we can talk about many other cell types in embryogenesis that behave like this, but the crest is a very good prototype. Crest cells will give rise to bones and cartilage and smooth muscle and neurons of so many different types, and glia and Schwann cells and endocrine derivatives. So is each cell able to give rise to the entire variety of derivatives at a given area of the axis? Or are we talking about fully specified progenitors, namely very early in development. Each progenitor, even before differentiating, knows what it is going to become. So these are crucial questions in development. Okay? So are we multipotent at the single cell level, or are we fully specified? Or is the crest a mixture of both type of progenitors, specified and multipotent? So to try and answer these questions, many people during the years did population versus clonal approaches. So population approaches are these grafts. And via these grafts, they could say that if I graft a very tiny segment of tube, still millions of cells, okay, I will get sensory ganglia and glia and sympathetics in a given place. So the immediate conclusion was that the crest is a multipotent structure, and this is still true, because at the level of the structure, at the level of the millions of cells, it is multipotent. But the question, the critical one, is whether at the single cell level a crestal is a committed cell. So this is something which was approached later by a clonal type of approach. So what is a clonal approach, and what does it test for? A clonal approach is very difficult to do in vivo. Also, I'm going to show you some things. A clonal approach is taking one single cell that you dissociate from a group of crest cells, and you put it in a dish. As a single cell, you put a base of fibroblasts or something to enable the cell to live, to proliferate, and you give it a medium, and then you come back many, many days later, and this cell has proliferated, it has divided, and it has created a clone. A clone is a group of cells derived from a single progenitor. And then you ask the question, is this clone composed of only neurons, or only glia, or only melanocyte, or is it composed of a mixture of cells? Now here, and we'll see the results in a minute, here you're not looking at lineage. And this is my point. You're not looking at normal development. You are looking at what we call potential. Namely, what is a cell able, and these are all very important developmental terms, what is a cell able to give rise to if I give it the best possible conditions in the world? Okay? The best possible conditions, which means FGF and NGF and EGF and all those factors which will enable you to survive, to proliferate, and etc. So this is not necessarily what the cell would have generated in vivo in an environment which is constrained by certain tissues, by certain factors and not others. This is what a cell is able potentially to give rise to. It's like a child. You said he has a lot of potential. He can become a musician, a physicist, and at the end he's going to be a truck driver. Okay? So his lineage is going to become a truck driver. But his potential when he was a child was absolutely wide and fantastic. Okay? So there is a very, very important distinction between potential and between actual lineage, what you do under normal conditions. Absolutely, you won't see the whole range. This is why science doesn't deal with the truth. Science never deals with truth. Science deals with what you can and are able to measure. Okay? So, this is one answer for clonal analysis. So what you can see here is that only 0.3% of the clones were able to show glia, neurons, melanocytes, fibroblasts, and cartilage. This was taken from the cranial crest. So each letter here means another type of derivative. So only 0.3% were what we call totipotent stem cells, namely generating cells which characterize a very multipotent progenitor. 80% generated what we called intermediate progenitors. Which is this? What is this? combinations of 3 to 4 different, or 2 to 4 different cell types, and different type of combinations. Glia melanocyte cartilage, glia cartilage, glia melanocytes, and so on. And this we call intermediate progenitors. And then 20%, which is a huge percentage, 20% generated what we called committed precursors, namely only cartilage or. What is the conclusion from this experiment? What can we conclude if this is what we get? We would, that it is a mixture with
You said before that it's not how it is really inside your body. Maybe the 0.3%, like the 30%, is not really exist in the body.
Absolutely, I totally agree. So what this term tells us about potential of the cells, is that even, it depends where you want to take it, is that even under conditions in which you basically can become most things, only 0.3% becomes most things. Most of them are intermediate, not surprisingly, and 20% are already committed to give rise to one single type of derivative. So this is as far as, and many experiments have been done in the 80s, 90s. Many people were concerned with these experiments. Now let's go to what happens in vivo. And these are data coming from our lab, and I'll show you a little bit of that. So what a guy in my lab, an excellent PhD student called Shlomi Crispin, did is we decided to follow the fate, not the potential, the fate of single cells and of groups of cells, no matter why, because we asked several questions, in normal embryos. And to this end, he developed a technique to inject DNA encoding for GFP into single progenitors in the dorsal tube, like that, just microinjection. And then he waited for 2 days and looked at the outcome. And I'm going to show you the animation and then a little bit of data. So what we found in these experiments is that basically there is a fate map of derivatives of the neural crest already present in the dorsal domain of the tube where the pre-migratory cohort of cells is sitting. With these early cells, the yellow cells that will become sympathetic neurons and glia, the cells which are located below, which will become Schwann cells, then DRGs, and finally melanocytes, and the last ones, the blue ones, will become the roof plate. They will close the neural tube. And this is a CNS population, a definitive CNS population. So look at the movie now, and you will see, I'm predicting because it goes fast. These are the first ones to leave, and they are going to go to the sympathetic ganglion primordium, which is here. Then the green ones are going to go to the nerve and become Schwann cells. The blue ones are going to be dorsal root ganglion cells. And finally, the red ones are going to migrate and give rise to melanocytes. So the first cell that comes out will have the longest journey, and then they will go on and on in a very stereotypic manner. Okay, so again, the yellow ones are going down, and then green and blue, and the red ones migrate underneath the ectoderm. And then the roof plate is formed as the definitive roof of the neural tube. So basically, the final homing site and the fate of the cells can be basically predicted from the localization of the cell in the dorsal domain of the tube, but also from the time it emigrates. Because this fate map is dynamic, it is a fate map in space and in time. So what Shlomi did at the beginning, where is this, was to take embryos at a certain stage and to map cells or mark cells here or here or here or here, namely at different dorsoventral positions in the tube. And then he did another exercise, which was the temporal fate map, which you can see here. I hope you can see. So this is how the single cell looks 6 hours after marking, because you need time for the DNA to be transcribed and translated into protein. So this is your 0 time. This is his single cell. And he did this exercise by marking only dorsal cells at different stages. So he did it in embryos at 25, having 25 somites, 27, 30, 35, very close stages. And he did it only at a very precise area of the axis, because we had to be extremely precise. And then what he finds is that if he did it at 25 somites, 2 days later, the green cells contributed to the sympathetic ganglia. You see the magnification here. If he did it a little bit later, they went to the Schwann cells. A little bit later, they contributed to the DRG, then to the melanocytes, and finally, they stayed and generated roof plate. These are not crest cells, these are presumptive roof plate cells. So there is a space and time can predict fate. So this means that there is no random behavior of the cells, even in such a multicellular animal. No random behavior of the cells. But this didn't tell us what.
Like what the reason, like what, if it's like encoded or if it's some chemical gradient?
Perfect. It didn't tell us whether this specification, this preciseness of the colonization of the target is due to an intrinsic map that the cells were committed, or due to the fact that they come out at different stages, and then the environment tells the cells what to become. So if I come very early, I have one pathway available, so no doubt I'm going to become a sympathoblast. But if I come out later, everything is already filled in. I have only one choice, to go subectodermally and become a pigment cell. So this type of results told us that there is a precise lineage coming out from different cells. So how would you test this idea of intrinsic versus extrinsic specification?
Take it out.
Yeah, so we don't want to take them out in vitro, because in vitro we don't test for fate. You can do something like that, because 2 days later it's not going to be the same cell.
Maybe there is a way to just delay make the cells like act slower and then.
Okay, so I'll give you one answer. There are several possibilities. I'll give you one of the answers, and this is what another excellent student at the lab did. So we know that neural progenitors, sympathoblasts, DRGs, etc., migrate dorsoventrally. But we know that melanocytes, which are the last ones to leave the tube, migrate under the ectoderm. So this is the ventral pathway, and this is the dorsolateral pathway. And we know that there is a receptor called endothelin receptor of type B2, EDNRB2, which is responsible for the dorsolateral migration of cells. So if we transfect neural progenitors, the first ones to come out, with EDNRB2, we will oblige these cells to migrate into the dorsolateral pathway, namely to migrate as melanocytes. And then we can ask, if we change the environment of the cells, do we transform them into melanocytes? Namely, does the environment determine the fate? So what happens based on your guess? So this is a complicated thing. The only thing I can tell you, I'm not going to show you this, is that with EDNRB2, like 40% of the cells migrate into the dorsolateral pathway. Okay? But when we analyze the outcome, you see them here with arrowheads, all of these cells did not become melanocytes. MC1, MELEM, c-KIT, all of them are markers of melanocytes. And these green cells are negative for the melanocyte markers. So somehow the environment was unable to reprogram the cells. But if we test for neural markers, all these cells migrating ectopically underneath the ectoderm express neural markers. So you can see here, for example, this green cell expresses Cash1, which is a marker for autonomic neurons. Here, these cells expressed islet1, these others expressed FOX, and so on and so forth. So it appears that these cells, even in an ectopic place, were able to upregulate markers which were neural, suggesting, and eventually 2 days later they die, because in the subectodermal space they cannot create synapses, they cannot fully differentiate. But this means that initial specification is independent of the migratory environment and basically depends on an intrinsic program, and I'm not going to show this to you, but okay, we know of several transcription factors that provide now a code, like the Hox code I told you last week, a code that determines neural versus melanocytic capabilities. And I'm going to skip some more fancy experiments that we did to show you data coming from the mouse. This is going to be the last 2 slides, in which we know that FOXD3 is a neural marker and is a progenitor marker. So what we did is we created a mutant for FOXD3 in the crest. FOXD3 is expressed by neural cells but not by melanocytes. And look what we see. What we see is that under control conditions, these are the melanocytes expressing the transcription factor microphthalmia, MITF, and you can see them coming out, and you can see them at a later stage migrating underneath the skin, the blue cells. Now, if we delete FOXD3, what do we get? Dorsal root ganglia in which neurons have been replaced by melanocytes. So we are able to reprogram them using this or one of the genes pertaining to the code that we discovered later on. And if it is a transcriptional factor code, means that this is an intrinsic type of decision, not depending on the environment, and this is genetics. Okay? So can you conclude for me? Can you wrap up everything regarding lineage decisions? How would you do that? Anybody? Doesn't have to be perfect. Just to know that you got the message.
Seems like different cells have different phases of that, and when we try to understand if these phases determined by external or internal factors, we realize that it's
Right. So at least the neural progenitors seem to be specified, or let me put it in a more accurate way, restricted. Okay? Why restricted? Because we know that single cells are still able to generate neurons and glia. Because we know that single cells are still able to generate sympathetic neurons, glia, and chromaffin cells. Okay? And because also we know that Schwann cell progenitors are also able to generate a certain type of melanocyte. So it appears that not all the cells are fully committed, some are fully committed, others are only restricted, and it appears that maybe others, in the head at least, are multipotent. So everything will depend on the developmental time and on the particular cell type that we are talking about. So everything gets extremely complicated, but the basic idea that in development everything is multipotent is wrong. There is order, and there are early fate restrictions among different cells. And this holds true not only for the crest, it holds true for different progenitors in the CNS as well. Okay, so these were the messages. I'm not going to continue, otherwise we should enter additional topics. Questions? Thank you so much.
הקשיות של האקסטרסלולר מטריקס?
יש כל מיני. אני לא יכולה להתייחס לזה, כי אני לא יודעת על איזה סטדיה את מדברת, אבל באופן עקרוני, הכל תלוי ב...
זה לא רק שהם יכולים... כאילו שזה לא רק הגנטיקה, אלא כן סיגנלים.
כן, יש הכל מכל, הכל קיים. יש לך את כל הווריאציה, יש המון תאים שהם מולטיפוטנטים, שהם ויכוח לגבי זה. כן? והכל עניין של תנאים, אבל שוב, אני שמה אותם בתרביל, הרבה יותר קל לקבל כל מיני דריבטים. כן, הרבה יותר קל. אפילו כמעט טריוויאלי. את משאירה אותם מספיק זמן ונותנת להם מספיק זמן. היי. כן, אני אשלח אותם למיקי והוא יעלה. תודה לך.
בסדר, אני לא יכולתי לענות לך. טוב, שלח לי.